Hi, this is Steven, and welcome to my YouTube channel. And today, we will be talking about one of the last processes in your central dogma of molecular biology, and we call it translation, the process of conversion of mRNA to proteins. But before that, don't forget to like and subscribe to my YouTube channel. Let's start. Before we'll start the translation process, we will be talking about the clinical significance of these translational control. It is very important that this translation process in your central dogma of molecular biology, or shall we say gene expression, is being talked about, because this is very critical for the development and survival of cancer. As you can remember in our cell cycle video, cells that are not committed to full development because they have abnormalities when it comes to their metabolism, when it comes to the vitality of the cells that are passing through the cell cycle that weren't really checked by the checkpoints, are growing abnormally inside your body. Okay, cancer cells must also frequently regulate the translation phase of your gene expression, though it is not fully understood why translation is targeted over steps like transcription. So in this process, you will see the photo on your left, a chest x-ray showing a lung cancer in the left lung. So cancer cells, often have genetically altered translation factors. These translation factors function to coordinate and catalyze the synthesis of polypeptides from mRNA by the ribosomes, or the protein synthesis machines inside your cell. And it is more common for cancer cells to modify the levels of existing translation factors. So these translation factors are altered, in your cancer cells, where they are being altered genetically. So this is a study done by Su in March 2020, the role of translation control in tumorigenesis and its therapeutic implications. Another clinical significance of studying translation are for your several major oncogenic signaling pathways that triggers the development of cancer cells, and that includes your RAS-MPK, PI3K, AKT-mTOR, the MYC, and this WNT beta-catenin pathways, and they ultimately reprogram the genome via translation. This is studied by Trott in 2016, the new frontiers in translational control of their cancer genome. So, for instance, your WNT beta-catenin pathways, are just one of the group of signal transduction pathways, which begins with proteins that pass signals into your cell through your cell membrane receptors. So these pathways are highly conserved. Evolutionarily, they are highly conserved in animals, which means that they are similar across animal species, from, let's say, for fruit flies until humans. And this WNT beta-catenin pathways was identified for its role in carcinogenesis, then for its function in embryonic development. This embryonic development process controls body axis patterning, cell fate specification, cell proliferation, and cell migration. And what you see in the background is a crystal protein structure of WNT8 and the cysteine-rich domain of frizzled A. And also, one of the clinical significance of studying translation is that cancer cells control translation to adapt cellular stress. They manipulate cellular stress, and this is through editing some part of the translation process, because during the stress, your cell translates mRNA that can mitigate the stress and promote survival. An example of this is the expression of your AMPK, or your AMP activated protein kinase, which is an enzyme, in various cancers. This activation triggers a cascade that can ultimately allow your cancer to escape apoptosis, programmed cell death. Okay, and this is triggered by nutrition deprivation. This AMPK is an enzyme which plays a big role in cellular energy homeostasis or balance. It also largely activating glucose and fatty acid uptake when your cellular energy is too low. And this is very important for future cancer therapies that involve disrupting the translation machinery of the cell to counter the downstream effects of cancer. Now let's talk about the translation process. Okay, it is a process by which the sequence of nucleotides in your mRNA direct the assembly of the correct sequence of amino acids in your corresponding polypeptide. Since it's in your genetic code lesson, you'll learn that the sequence of amino acids are non-overlapping, okay, or your genetic code are non-overlapping, and they are degenerate. So what is the correct sequence of your polypeptide or amino acids will be reflecting on the sequence of your mRNA and their nucleotides. It takes place on ribosomes, and it coordinates the movements of transfer tRNA, which carries specific amino acids with the genetic instructions of an mRNA. And as we examine your cell's translation machinery, we first describe the structure and function of tRNAs and your ribosomes, and we then explain how these components interact during translation. Let's see the overview of your animal cell, or the diagram of typical animal cells. So you'll see here we have the nucleolus, the nucleus, and this is your rough endoplasmic reticulum because they are studied with ribosomes. And these number 3 here are your ribosomes. They are the sites of protein synthesis. Okay, ribosomes could run through or be studied in your endoplasmic reticulum, or they could be free floating in your cytosol or your cytoplasm. So this is a process of your translation. Okay, the overview of this process, this animation is the elongation and membrane targeting in your eukaryotic translation. So the ribosome is in green and yellow. We have the large and the small subunits. The tRNAs are in dark blue, okay, and the others are in light blue. The produced polypeptide is released into your endoplasmic reticulum, and the shape of your ribosome is computed with something like quantum models. Okay, so this is the overview of the process, and they are following also with this process where there is a tRNA bound to an amino acid, which is phenylalanine. Okay, it carries an anticodon, and once it reads the messenger RNA, okay, it will be attached to the ribosome, and all of the growing peptide chains will be interconnected with each other, because there is the nitrogen part of your phenylalanine will just grab the carboxyl part or the carbon part of your aspartic acid. Okay, so this is your protein synthesis and in overview. Let's talk about tRNAs, or what we call as transfer RNAs. tRNAs mediate the translation of mRNA codons to amino acids. So there is no obvious chemical similarity or affinity existing between the nucleotide triplets of mRNA codons and the amino acids they specify. Rather, tRNAs serve as adapter molecules that mediate the transfer of information from nucleic acids to proteins. So let's talk about the structure of your tRNAs. There are very short, single-stranded RNA molecules with 74 to 95 nucleotides in length. And several nucleotides in tRNAs contain modified bases that are produced by chemical alterations of your principal A, G, C, and U nucleotides. So let's see the tertiary structure of your tRNAs. It has your CCA tail. Okay, by the way, this is your tRNA phenylalanine from yeast. Okay, this is an X-ray structure. This is a CCA tail in yellow. Okay, an acceptor stem in purple, a variable loop in orange, a D-arm in this one, a D-arm in blue, this with anticodon in black, this is your anticodon, and T-arm in green. So it has 3 arms, the D-arm, your anticodon arm, and your T-arm. And tRNAs, again, has modified bases and produced by the chemical alterations of your adenosine, cytidine, uridine, and guanosine. These are the normal bases. And then the modified bases of uridine are your ribothiamidine, the dihydrouridine, pseudouridine, 4-thiouridine. In your cytidine, we have 3-methyl cytidine and 5-methyl cytidine. The adenosine, we have the inosine, N6-methyl adenosine, and 1-methyl inosine. And for guanosine, examples of them, some of them are 7-methyl guanosine and your queunosine. We also have the other one here. And one of the primary structures of tRNA molecules has the same thing with your mRNA. It has a 5' because they are single-stranded, 5' to 3' ends. Okay, they fold to form a characteristic of secondary and tertiary structures. Just like your proteins, they have secondary and tertiary structures. The anticodon and the amino acid attachment site are at the opposite ends of your L-shaped structure. This is your 5'. This is your yeast tRNA of alanine. It has 5' and 3', and this is your amino acid attachment site. This is where the amino acids are attaching, forming a polypeptide chain. Okay, these are your anticodons, and this is your mRNA codons. These are the arms. Okay, as shown earlier from the previous slide. It is possible to consider the structure of tRNA on 3 levels. First, the nucleotide sequence of a tRNA will constitute a primary structure, this one. The second, a short complementary regions within tRNA single strand can form base pairs with each other and creating a characteristic of a clover leaf shape. And this is your tRNA secondary structure. And lastly, folding a 3-dimensional space creates a tertiary structures that looks like a compact letter L, and this is your tertiary structure. So we know that there are enzymes, okay, which we call as your amino acyl tRNA synthetases, okay, which catalyze the attachments of tRNAs to their corresponding amino acids. At the other end of your L, where the 5' and 3' ends of the tRNA strands are found, there are enzymes known as your amino acyl tRNA synthetases, which connect the tRNA to the amino acid that corresponds to that anticodon. So what happens during this process? The amino acyl tRNA synthetase will activate your amino acid. In this instance, this is your glycine. Okay. Then the enzyme will transfer the amino acid's carboxyl group from AMP, okay, to the hydroxyl group of the ribose at the 3' end of the tRNA, which is producing a charged tRNA. So you'll see here, it transfers its phosphate group to glycine, okay, to adenosine triphosphate, and then the synthase, your tRNA synthetase will cleave off a charged tRNA, okay, carrying the amino acid. So this is technically phosphorylation from ATP to adenosine monophosphate. So what are the critical role of your base pairs between codons and anticodons? You'll see on the structure that the tRNA carries the anticodons and the codons are within the mRNA region. So this attachment of an appropriate amino acid will charge a tRNA. The amino acid itself does not play a role in determining where it becomes incorporated in a growing polypeptide chain. Instead, it's the specific interaction between the tRNA's anticodon and mRNA's codon will make that decision. So you'll see here a base pair between an mRNA codon and a tRNA anticodon, which determines the amino acid which is added to a growing polypeptide. So your tRNA for cysteine anticodon is ACA. Then the opposite for that codon will be UGU. Thus, it carries cysteine, okay, on this amino acid attaching end of the 3' end of your tRNA. So they are treated with nickel hydride, okay, to change the amino acid. So the cysteine became alanine, however, the anticodons remained unchanged once they are treated with nickel hydride. So what does it imply? It implies that the codons and anticodons are specific, okay, are well coordinated on how the tRNA will produce or on what amino acids that are being carried by your tRNA, okay, for this growing polypeptide. So let us talk about the question, how can an organism construct proper polypeptides if some of the codons in its mRNAs cannot locate tRNA? So let's say if the mRNAs are having a hard time to locate the tRNAs with the complementary anticodons. The answer is that some tRNAs can recognize more than one codon. Okay, for the amino acids they were charged. That's the degeneracy of the code. There are a lot of codons will specify only one amino acid. That is, the anticodons of these tRNAs can interact with more than one codon for the same amino acids, in keeping the degenerate nature again of your genetic code. Although researchers don't fully understand this promiscuous base pairing between codons and anticodons, Francis Crick spelled out a few rules that govern it. And this is what we call as your Wobble. Okay? One tRNA, and they will be classifying more than one codon, okay, or reading more than one codon. So the flexibility in base pairing between your 3' nucleotide in the codon and the 5' nucleotide in the anticodon is known as Wobble. Okay, these are the Wobble pairs for inosine and guanine. This is your inosine, cytosine, inosine, uracil, inosine, adenine, and guanine, uracil. These are the Wobble, or these are specialized pairs. They do not follow, again, your Watson-Crick base pairs ruling. Again, these are the four main Wobble base pairs: the guanine uracil, hypoxanthine or the inosine, okay, hypoxanthine or your inosine and adenine, and inosine and cytosine. So this is using inosine, okay, instead of hypoxanthine. That is to maintain the consistency of your nucleic acid nomenclature. This is your the G at the 5' end of your anticodon, and showing that it could pair with either U or C. This is your 5' anticodon, okay, and in this mRNA codon, The wobbles are following a specific rule, okay, according to um, certain scientists that discovered the wobble, okay. The G part here of your anticodon 5' end can either pair with U or C, okay, in the 3' end of your mRNA codon. So let's talk about the wobble rules, okay, the pairing possibilities for other nucleotides at the 5' end of the anticodon and the 3' end of the anticodon. So we have this GCAUI. Okay, these are the nucleotides on the 5' end of the anticodon, and they can pair with the 3' end of the codon. It's either for G it's U or C, for C again it's G, for A it's U, for U it's A or G, and for inosine it could be U, C, or A. This refers to the observation that nucleotide at the 5' position of the tRNA anticodons can often pair with different nucleotides at the 3' position of your mRNA codon, and this explains why alternative codons for a single amino acid usually start with the same nucleotides. And this is very important. This wobble concept is very important, so that the degeneracy of the code in your degeneracy of codes, okay, is highly conserved in the translation process. Let's talk about the organelles or subcellular structures in your cell that are the sites of polypeptide synthesis, or the ribosomes. Okay. The ribosomes is facilitating your polypeptide synthesis in different ways. First, they recognize mRNA features that signal the start of translation. Second, they help ensure accurate interpretation of the genetic code by stabilizing interactions between tRNAs and mRNAs. And without a ribosome, codon and anticodon recognition, okay, is mediated only by 3 base pairs and would be extremely very weak. And lastly, they supply enzymatic activity that links the amino acids in a growing polypeptide chain. Fourth, by moving 5' to 3' along an mRNA molecule, they expose the mRNA codons in sequence and ensuring that there is linear addition of amino acids. And finally, ribosomes help end polypeptide synthesis by dissociating both the mRNA and directing polypeptide construction and from the polypeptide product itself. So they also help to stop the polypeptide synthesis, okay, through dissociation and stopping the connection between the mRNA, okay, mRNA that directs polypeptide creation and the product production itself. So the structure of ribosomes is, for example, this is for your E. coli. There have 3 ribosomal RNAs or rRNAs and 52 different ribosomal proteins. A photo or micrograph on your right is an SEM micrograph artificially of colored E. coli colonies. Let's take a look at it. The structure of your E. coli 70S ribosome. This is a large subunit, okay, in red, and the small subunit in blue. So these are just photos, okay, measured in amperes. The scale bar is for amperes. So the ribosome has subunits, okay, which is the small and the big subunit, or large and small subunits, and they're composed of RNA and protein. For the prokaryotic cells and the eukaryotic, they differ in this the size, okay. For the eukaryotic, it's quite larger, it's 80S, and for prokaryotic, it's 70S. The subunits for the prokaryotic is 50-30, while in eukaryotic, it's 60-40. The number of proteins, okay, we have here 31, 21, 45, 33, and the nucleotides available. So this is the prokaryotic ribosome and the eukaryotic ribosome. For the prokaryotic cells, very important clinical significance of this ribosomes is that the target, okay, of your antibiotics, let's say for example for your aminoglycosides, Okay, these antibiotics target the 30S. Okay, your macrolides or the tetracyclines, erythromycins targets the 50S. They have this rRNAs within, okay, ribosomal RNAs within your small and large subunits, and that is the target of your antibiotics. They destroy the protein creators of your bacterial cell. Again, a number of antibiotics acts by inhibiting translation, and they include anisomycin, cyclohexamide, chloramphenicol, tetracycline, streptomycin, erythromycin, and pyromycin. These are the top three antibiotics shown in your right, because prokaryotic ribosomes have a different structure from that of your eukaryotic ribosomes, and thus antibiotics can specifically target bacterial infections without harming any of your eukaryotic host cells. And for the different parts of a ribosome, we have different functions. This is your peptidyltransferase, large subunit and small subunits, your aminoacyl site, peptidyl site, and your exit sites. Okay, the small subunit initially binds to your mRNA. Okay, your large subunit has an enzyme, peptidyltransferase, okay, which catalyzes the formation of your peptide bonds. They hold the peptide bonds until it reaches a very long chain. This is a ribosome of atomic structure of your 30S subunit of your thermopilus, okay, a gram-negative bacterium, which is very useful in a large range of biotechnology applications, like its model organism, genetic manipulation, and in your structural genomics. It's very useful. Okay. Another large subunit of a bacterial ribosome. This is shown. The ribosomal proteins are shown in lavender. Okay. The rRNA is in gold and white, and the 5S rRNA is shown in maroon and white. This is quite complicated. This is done through X-ray crystallography and elegant techniques of electron microscopy. They were able to gain a remarkably detailed view of the complicated structure of the ribosome. So this is your X-ray crystallography. Okay. Another sketch is coming from this structure. We have the P, A, and E sites. Okay. This is your growing newly born proteins. This is your large subunit. This is your small subunits. This is your mRNA strand. Okay. And these are your tRNAs carrying the amino acids. So let's talk about different processes of translations in ribosomes in your prokaryotic versus in eukaryotic cell. We have what we call as your initiation phase. Okay, the same thing with transcription. It is more differentiated from prokaryotes and eukaryotes. In prokaryotes, okay, we have this particular part here. We call this the Shine-Dalgarno box, okay, which is a sequence of amino acids, AGG. Okay, it has this particular nucleotides, and there is an initiating codon, or the AUG. Okay, this AUG is carrying your F-methionine. Okay. In eukaryotes, it is the methionine, but for the prokaryotes, it's the F-methionine. So what's special about the prokaryotes is that they have the Shine-Dalgarno box, they have the initiating codon of F-met, okay, and they passes through this large subunit of the 50S of your ribosome, and it reads in the P site, okay, the peptidyl site. However, in your eukaryotic cell, it must have this methylated mRNA cap. Okay, the methylated mRNA cap, and there is what we call as an untranslated leader. before it starts with an initiating codon. Okay, it scans through, it will read the same process, it will pass through the large and small subunits. Anticodon of your tRNA will read, okay, the codon found in your mRNA, then it will be reading the methionine. So the next process is what we call as elongation. Okay, this is an addition of amino acids to growing polypeptide, and the protein known here is what we call as the elongation factors. Although not shown, it ushers the appropriate tRNA into the A site of the ribosome, or the active site of the ribosome. The anticodon of this charged tRNA must recognize the next codon in the mRNA. So the ribosome simultaneously holds the initiating RNA at its P site and the second tRNA at its A site, so that the peptidyl transferase can catalyze the formations of the peptide bonds between amino acids, which are carried by the two tRNAs. And as a result, the tRNA at the A site now carries two amino acids, and the N terminus of this dipeptide is F-met, and the C terminus of the second amino acid, which carboxyl group, remains covalently bound or linked to its tRNA. This is a very simple process, as we showed in this figure. Okay, this elongation phase, this phenylalanine, okay, will grab, okay, the F-met, okay, once it reaches your P site. So this phenylalanine will just pull your F-met, okay, and it moves forward to your P site. Thus, the tRNA that is being left, or being disassociated with the amino acids, will enter the E site, and then they will be soon disintegrate away from the ribosome. Okay, again, the ribosome moves towards the 3' end of the mRNA during this step. This is the direction of your ribosome movement. So they move from 5' to 3'. Okay, so there is what we call a polyribosome, okay, where they simultaneously translates the same mRNA, okay, which although they grow the polypeptides, okay, some subunits are being released, okay, once they are done with their process, and this is complex mechanism is allowing simultaneous synthesis of many copies of a polypeptide coming from a single mRNA. This is the elongation phase. It goes on and on, okay, and once it reaches the final stage, this termination stage, the ribosome then releases the complete polypeptide. So no normal tRNAs carry anticodons complementary to the three nonsense stop codons, your UAG, UA, and UGA, UAG, UAA, and UAGA. Thus, the movement of this ribosome brings a nonsense codon into the ribosome's A site. This is your A site. Okay, it has the tRNA has to bind, okay, to the termination codon, and proteins called release factors will recognize the termination codons and bring polypeptide synthesis into halt. Okay, the tRNA specifying the C terminal amino acids will release, okay, this completed polypeptide chain, and the same tRNA as well as the mRNA will separate or disintegrate from the ribosome, and the ribosome will dissociate from its large and small subunits. So this is free floating, okay, in your cytoplasm. Then they will be digested by the lysosomes of your cell. Take note that there is also processing after translation that will change the polypeptide structure. So protein structure is not irrevocably fixed at the completion of translation. There are several processes that may subsequently modify a polypeptide structure. So that structure that you found from the termination stage would be modified along the way. Cleavage may remove amino acids such as the N-terminal F-met from a polypeptide, or it may generate several smaller polypeptides from one larger product of translation. In the latter case, the larger polypeptide made before it is cleaved into small polypeptides and often called as polyproteins. Polyproteins are chains of covalently conjoined smaller proteins that occur in nature as very versatile, and it means this example of it will be the proteome of some of the viruses, including your HIV-1 virus. This is your HIV virus. It has proteases here. Then this is creating the proteome. These are the proteins of your HIV. Processing after translation also can change your polypeptide structure, and there are some, again, cleavages and additions which are known as your post-translational modifications. These post-translational changes to a protein can be very important. For example, the biochemical function of many enzymes directly depends on the addition or maybe sometimes removal of different phosphate groups. And for instance, this modification of your insulin, okay, at the top you'll see here, the ribosome is translating an mRNA sequence into a protein, okay, this one, and insulin is passes the protein through the endoplasmic reticulum, okay, it passes through this, it folds, oxidizes, and signal peptide cleavage, okay, and this is where it is being cut, okay, using the Golgi transport and vesicle packaging. Then the protein passes through the Golgi apparatus where it is packaged and secured into a vesicle. Okay, in the vesicle, more parts are being cut off, and it turns into mature insulin. So this process is quite complex, but you will just take note that insulin is undergoing this extensive post-translational modifications along this production pathway. Okay, production and secretion is really important and largely dependent on the prepared insulin that is awaited for secretion. So both the C-peptides and the mature insulin are biochemically or biologically active, and cells components and proteins are not scaled in this image. Okay, so this is your post-translational modification of insulin, and this is some of the processes that may happen into an amino acid when it's being undergoing a cleavage. Okay, it's removing an amino acids from the N-terminus of a polypeptide, or split a larger polypeptide into smaller functional proteins. So this one is removing the F-met, and this is now the new N-terminus of your polypeptide chain. It could be splitting a polyprotein from this long chunk, okay, into multiple smaller polypeptides. It could be adding chemical constituents that will modify a protein. Let's say for serine, a phosphate group is being added to serine, and then there is a process of phosphorylation that is being added, okay, in this polypeptide chain. And lastly, we have what we call as post-translational modifications. Okay, again, it could be adding, like glycolysation, adding glycogen. It is very important for some of the antigens, your red blood cells, A, B, Rh, okay, blood typing. For lipids, addition of lipids, that is important for the architecture of your cell membranes or the organelle membranes. Phosphorylation, very important for activation or inactivating of protein, inactivation of proteins, your protein kinases and your CDKs during this cell cycle. Okay, you remember your cell cycle has checkpoints, and these are this elevate or suppresses their amounts during this process. Hydroxylation is very important for the creation of collagen in your body. Okay, collagen synthesis. 5, it's methylation and acetylation. So, for instance, if you add a methyl group for the histone, it will decrease the transcription. If you add acetyl to the histone group of your DNA, it will increase transcription. And lastly, trimming, okay, for an active to inactive form. For trypsinogen, inactive and its inactive form, and will become trypsin for the active form of these digestive enzymes. So that's it. That's all we have for today. Thank you for listening the translation process, and I hope you've learned something today. Thank you and goodbye.